who is Professor Hank Greely, who directs the Center for Law and Biosciences.
Law Biosciences.
Law and the Biosciences.
Yeah, we have an article.
Stanford. You will notice that the name is not quite the same as ours. Ours is the Center for Law and Biomedical Sciences. We didn't want to steal their name. On the other hand, we learned a great
Could
deal.
have rented it.
License.
But they are incredible leaders in this field. Professor Greely is the Dean F. and Kate Edelman Johnston Professor of Law at Stanford University. He does a tremendous amount of work in law and neuroscience and law and genetics. And he's going to...
And any other bright, shiny objects that cross my visual field. Yeah.
Yes, well, and now I'm going to get out of the way so you
Okay.
can share something.
So the most important thing that our Center for Law and the Biosciences does is turn out great law professors, like your own Tennille Brown, who is a great deal because we claim her as our fellow, although I never actually had to pay for her.
I was paid, but not by you.
But not by me. Though I did listen to you a lot.
Yes.
So there's some cost involved. So it's great to be here. I want to talk about CRISPR, but I want to start by talking about what I think is probably the most consequential invention of the 20th century. It's the Model T Ford. Model T was important not because it was the first car, because it wasn't. There had been cars for 20, 25 years. Kind of when the first car was is one of these definitional issues that's debated. But it was important because before the Model T, cars were expensive. They broke down all the time. You needed to hire a full-time chauffeur/mechanic if you wanted your car to run, kind of like the Irish guy in Downton Abbey. And as a result, you only had a car if you were rich. And as a result of that, there weren't very many cars. Comes the Model T, you've got a car that is cheap, that is durable, that runs forever, and that anyone can use and anyone can afford. I actually should go back and see if I could find the numbers at some point, but my guess is in California before the Model T there were maybe 5,000 cars, and 10 years after the Model T there were probably 200,000 cars. There are now over 20 million cars in California. The reason I bring this up is it is often the case that the most important step is not the first step. I mean, you can't do any of the other steps without the first step, but using that logic, the Big Bang caused everything else, and that was the most important thing, and we can end all discussions quickly. For cars and all the changes they've made to how we live, how our cities are laid out, the air we breathe, the temperature of the air we breathe, all the ways cars have revolutionized our lives, if they were still extraordinarily expensive, those wouldn't have happened, and not nearly as much as they have. So now, to answer the question that's going through some of your minds of, I didn't think I was coming to a seminar on cars, what's it got to do with CRISPR? CRISPR is the Model T of genetic engineering. You had been able to do genome engineering, DNA editing before CRISPR. Actually, arguably the very first person to do it was my friend and mentor Paul Berg at Stanford, when he used various phages to move a DNA sequence from one bacterium into another bacterium and invented recombinant DNA, for which he won the Nobel Prize. That was back in about 1971 or so. But it didn't do very much. It didn't do it in a very targeted way. It wasn't very precise. It was expensive and difficult. More recently, there have been a couple of really very good systems of doing gene editing. TALENS, which stands for something, it's T-A-L-E-N-S, T-A-L-E-N, and then the S is the plural somehow, and zinc finger nucleases. And they work really well. In fact, there's an argument that maybe they even work better than CRISPR, but they're not cheap and easy to do. To do one takes months and months of work. To come up with a new zinc finger nuclease or a new TALENS that will precisely edit a specific sequence of DNA takes months of work, highly skilled work, postdocs, time, money. It's several $100,000 in several months to come up with something that might work or might not. Ultimately, you'll probably get it to work, but it may take more time and money. I remember deciding that CRISPR was actually going to be a big deal when I talked to a friend of mine at Stanford Medical School, and I said, so has your lab started using CRISPR yet? And he said, yeah, we have. I said, so how easy was it to use? And he said, well, I figured it would, I heard everybody was telling me it was wonderfully easy. I figured take my lab a month or two to figure out how to do it, to get it up and running. Turned out to be an afternoon. That's the change. It goes from expensive, difficult, requiring lots of high-skilled labor, lots of time, lots of money, to cheap and easy. A kid with a bachelor's degree in biology and $1,000 and a garage could start doing CRISPR. I know somebody who for I think it's now $199, will sell you do-it-yourself CRISPR kit. Got started on, I think, Indiegogo or one of those self-funding things. They're selling do-it-yourself CRISPR. Now, they're selling you the bacteria and they're selling you the specific guide RNA, so it's not sort of generic CRISPR. But the point of it is, it's cheap and easy, it's accurate, it's not perfectly accurate yet, but it's accurate, and it's democratic. Anybody can do it. And all those are good things, like cars are good things. And all those are bad things, like cars are bad things. That kill 35,000 Americans every year, and that pollute the atmosphere, and that make cities less livable. But as a Californian, of course, I'm a human, non-human chimera. I'm half human and half car. So all revolutions in science, I actually believe this is literally true. Except maybe for general relativity, are revolutions in tools. Astronomy changes when you've got telescopes. Microbiology changes when you've got microscopes. Sometimes the tools aren't physical tools. Science changed when either Leibniz or Newton, depending on who you talk to, invented calculus. Whether you're talking to a Brit or whether you're talking to a German. Computers are tools, machine learning algorithms are tools that are changing science and changing the world. CRISPR is such a tool. I believe that CRISPR is going to turn out to be the most revolutionary tool in biology in many decades. I think the only possible competitors right now are things like PCR, polymerase chain reaction, and maybe green fluorescent protein and the other protein markers. Two things, by the way, that were adopted almost immediately everywhere because they were cheap, easy, and transformative tools and allowed you to do a whole bunch of stuff easily that you couldn't do before very easily. CRISPR falls into that category. CRISPR is really intriguing also because every month, it seems, I see somebody who's come up with a new way to use CRISPR. And it's the basic CRISPR idea is a scissors. But then you cut and you put something else in. And now I just read something the other day about people using CRISPR to change the epigenetic markings of some DNA. They weren't changing the sequence of the somatic DNA. They were changing the epigenetics of it, which caused something to be expressed that wasn't otherwise expressed, that has great potential for use in medicine. So CRISPR is a big deal. What I want to talk about after that short commercial for CRISPR, and no, I get no royalties, alas, although I do know both Jennifer Doudna and Feng Zhang and Emmanuel Charpentier, and some of them are likely to get royalties, although who is not entirely clear. I want to talk about two things, maybe three if we have time. First, what we are worrying too much about with respect to CRISPR. Second, what we are not worrying enough about with respect to CRISPR. And third, if we have time, what should be deeply embarrassing to all those involved, the CRISPR patent fight. So let me start with what we're worrying too much about. And I can claim to be, having been in near the beginning of this, in January of 2015, in October I was invited, and in January I went to a small meeting at a resort in Napa Valley that Jennifer Doudna called. Jennifer is probably the person who is most often given most credit for CRISPR, although like all important inventions, success has 1,000 parents and failure has none. But Jennifer, I think, probably deserves to be viewed as the most important and is certainly one of the most important. She became concerned about the ethical implications of CRISPR. And she actually tells the story that showed up in a New Yorker article by Michael Spector of what caused her to really get concerned. She had a dream. Her friend, who's actually, I think, here at Utah, Dana Carroll, came to and said, I've got a man who wants to talk to you about CRISPR. He really is very, very interested, so she begins to explain it to this guy, but his back is turned to her. And when he turns around, it's Hitler with a pig snout.
Oh my
I
God.
don't know why the pig snout was on it. I don't know whether there's a
It's
mustache in the
anti-Semitic.
pig snout, but it was Hitler. And she woke up in a cold sweat. And she said, I really need to think about this some more. So there were about 14 of us around the table, 12 scientists, me and one other law professor, Alta Charo from the University of Wisconsin, who's done law policy work in biosciences for years and years. And we spent a day discussing the ethics of CRISPR. It was, for me, historically very interestingly significant. It took place 1 week before the 40th anniversary of the Asilomar recombinant DNA meeting, which was the very famous meeting in which a bunch of scientists got together and declared a moratorium on doing recombinant DNA work until they were more confident that it could be done safely. They weren't actually worried about the ethical issues at that point. They were worried more about safety issues. But it's written up in it's a very famous meeting. It had taken place exactly 40 years before a week later. And furthermore, two of the people at this table at Napa were Paul Berg and David Baltimore, both interesting people, both Nobel Prize winners, and two of the 5 people on the organizing committee for the Asilomar meeting. So I had a sense of history, maybe not repeating itself, but at least rhyming. We talked for 7 or 8 hours around the table. Alta and I, the 2 lawyers, which may be significant, kept saying, let's talk more about non-human uses. But nobody else wanted to talk about non-human uses. And even in the human uses, nobody really wanted to talk about the somatic cell uses, the uses that will only affect your body while you're alive. They wanted to talk about the germline uses. And it is those germline uses of CRISPR that I think we have already spent way too much time and attention on. They are worth paying attention to, but they are not urgent. They are decades away. And I believe we will benefit from spending those decades seeing what happens with CRISPR, seeing what happens in the rest of the reproductive technology world before we make any firm decisions about how to go forward. I do agree with the position that this group reached, which was then published in Science in late March of 2015, calling for a moratorium on any germline modification of humans. But it was a moratorium. A moratorium is not a ban. A lot of people read moratorium and say ban. A moratorium in theory is something that can be lifted. It is a let's stop and look around. It's a timeout. It's a temporary restraining order. It's something that says, let's not do it right now, let's think about it some more. So that's what our group called for. Our group also called for more discussion, and in December of 2015, the National Academy of Sciences, the Chinese Academy of Sciences, the Royal Society of the UK held the great CRISPR gala the great CRISPR circus of December 2015 at the National Academy's building, a 3-day meeting with literally a cast of scores, probably more than 100. I was one of the many speakers at that. To discuss this, there were 4 of us speakers who mentioned non-human uses. So the focus has been strongly on human uses. It's been very strongly on human germline uses. In fact, a week before our paper came out in Science, a paper in I forget whether it was the first author or the last author, was a guy named Ed Lampierre, who was then the CEO of a company called Sangamo, published a paper in Nature saying basically, I'm a law professor, maybe I'm a little cynical, saying somatic uses, which my company is doing, good. Germline uses, which my company isn't doing, bad. Let's kill all the germline uses, says the guy doing the somatic uses, and make sure, we're the good guys. We're the good guys, they're the bad guys. We're the good guys, they're the bad guys. That paper came out. A week later, our paper came out saying moratorium. I support the moratorium mainly on safety grounds, but even beyond safety grounds, I think it's useful for us to stop and think about some of the larger ethical questions. But I think it's ultimately an open issue. What came out of the December 2015 meeting was a statement by the organizing committee, which was chaired by David Baltimore, and I hope you all get a chance at some point in your life to meet David Baltimore. He's an interesting guy. He is not meek. And basically what David wants usually happens. Bull in the china shop is probably not quite strong enough. Well, no, that implies he's breaking things. But David is strong-willed and often gets his way. And so the organizing committee issued a statement that said what David wanted it to say. And what David also wanted was almost the entire press confuse the organizing committee with the National Academy of Sciences. And so the National Academy says X, driving the National Academy crazy because it hadn't said it. The committee that had organized this particular meeting had said it. The National Academy then appointed an expert committee. The National Academy is a funky organization. It's now the National Academies of Science, Engineering, and Medicine. And it's partially an honorary society, and it would be a great honor to be named to any of those. But it's also a really expensive think tank. And if they can find people who will pay them $1 million, they will give them a really, really good, detailed, lengthy, thoughtful report on an issue. Usually it's the federal government that pays them, but not always. So they set up a committee to study this. That committee reported, and Alta Chara, who was one of the co-chairs of it, and is a friend of mine, swears this is purely coincidence, reported on Valentine's Day of last year about human uses of genome editing. They took it into 3 pieces. They said in vitro uses, uses in the lab, uses in petri dishes, shouldn't worry about hardly at all. Normal sort of biosafety things, but nothing special. Somatic cell uses, things where, let's say, Tennille had a genetic disease and we use CRISPR to snip the offending parts out of the relevant genes and add the good parts, add functional parts in. Those make Tennille healthier, but they die with her. They don't get passed on to her kids. They're in her lung cells or in her skin cells or in her liver cells. They're not in her eggs. So those somatic uses, this committee said, full speed ahead with this, full speed ahead in the context of the FDA and other safety sorts of regulations. And then they got to germline modification. And there they said, stop and think and discuss, but they didn't call for a complete ban. They said there may be circumstances where if this is proven safe, this is useful. And that's basically my position as well. Although it was my position before the National Academy came out with it, though I couldn't claim IP rights and sue them over it. And it's my position for several reasons. First, we have a lot of time to deal with CRISPR babies because this is not going to happen soon. The safety kinds of testing that would be required will take at least a decade, assuming that companies are allowed to do the safety testing and assuming that the FDA is allowed to consider the safety testing. The FDA has asserted, and I think legally so, that it has jurisdiction over CRISPR babies on the grounds that it involves cells that are more than minimally manipulated, that thereby become biological products or drugs. I think it's odd to have a human embryo be a drug, but under FDA statutes and interpretations, a human embryo whose cells had been more than minimally manipulated would be a drug. So under current US law, and law almost every place else, you need safety approvals. That's going to take a long time. You also need the political situation to allow this to go forward. And even without formal total bans, which do exist in a number of countries, there are other things going on. In the United States right now, this is legal to do if you have FDA permission, either as a biological license approval or a new drug application, a new drug approval, or you have permission to do it as research under an investigative new drug permission. But to do that, you got to file something with the FDA. Congress added to the last appropriations rider the last two appropriations bills, not appropriations, continuing resolutions, and I am sure it's in the one that was signed over the weekend, a provision that says FDA can spend no money considering any issues having to do with, any applications having to do with human germline modification. I unfortunately think I may be responsible for turning that, I mean, I actually agree with it for right now, but I may be responsible for making that effective. Because when they first proposed that language, I was quoted in some press as saying, those morons, I don't think I was quoted as morons, but that's what I think I said to the reporter. Don't they realize when you file an IND, a request for an investigational new drug exemption, if the FDA does nothing, it becomes effective within 60 days? So if you ban the FDA from spending any money on it, the FDA can't block it, and therefore it becomes effective. So not only does it say the FDA can't spend any money on it, they say, no such application shall be deemed to be received. I love that. You can have the entire Supreme Court witnessing the fact that you handed the application in at the FDA, and it doesn't matter, because it's deemed not to be received. That's a year-at-a-time provision. Of course, the prohibition on federal funding for human embryo experimentation is also a year-by-year appropriations bill rider that's been in every appropriations bill since 1995. So it has to come up, has to be redone every year, but it may well be redone every year. So you've got these safety barriers, you've got political barriers to even getting to the safety issues. But I think there's an even more important issue. For CRISPR babies, there's no killer app. Sorry, I'm from Silicon Valley. In this context, that sounds a little tough, but that's the language they use in my bubble. There's no good reason to do it. Almost no good reason to do it. If you want to do it to treat disease, genetic disease, almost every genetic disease that could be treated by successfully editing the genome of the embryo, more likely they'd edit the genomes of cell lines that give rise to eggs and sperm rather than edit the embryo for reasons I can go into later. It's safer and more controllable that way. Every disease you can take care of that way, almost you can take care of by embryo selection. So rather than edit an embryo, you take 4 embryos and you look for the ones that don't have the disease. How many of you know that this is possible? Okay, you're a relatively sophisticated crowd. Most times it's like 10 to 15% of people know that this is not science fiction. Not only is it science fiction, any of you students could be the world's first baby who was born as a result of this process, because that kid is now 27 years old. Maybe 28, was born in 1990, but I don't know when in 1990. This is a process called pre-implantation genetic diagnosis. You take an embryo when it's earlier than 5 days old. You take one or several cells off it. You take one cell if it's a 3-day embryo with about 8 cells, which is how it started to be done. Now you take 5 or 10 cells off a 5-day embryo, which is a blastocyst, which has several hundred cells, and you test those cells. And you look to see, okay, both of you are carriers for cystic fibrosis. That means on average, 1 in 4 of your children will get 2 copies of the harmful version, the harmful allele, the harmful genetic variation, and 3/4 of them, and that 1/4 would get the disease, and the other 3/4 won't get the disease. Half will be carriers, 1/4 of them will not even be carriers. Those are statistics. In any individual case, your mileage may vary. You could have all 4 of them be affected, you could have all 4 of them be completely clean, but on average, over time, 1/4 affected, 1/4 completely normal type, half mixed, carriers who don't themselves get the disease. And you can use that and pick out the embryos that are safe. And that's a procedure that's been done for 27 years. That's offered by hundreds of fertility clinics in the United States. That is not exactly FDA approved, but the parts of it that needed approval are approved. And then we know is relatively safe. Basically as safe as IVF. None of those kids are born missing an arm, a leg, a head, or other appendage because of the cell that was taken off. The other cells make up for it. There are only really 3 situations I've been able to think of where PGD would not be sufficient. Leaving aside, I mean, there's the possibility that you only have 1 embryo or 2 embryos and they're both affected, to which I would say make more embryos. Go through egg harvest again and sperm harvest again and make more embryos. But genetically, there are only 3 situations where this wouldn't work, as far as I can tell. First, and this is likely to be the most common, if both prospective parents have what we call an autosomal recessive disorder, and the same autosomal recessive disorder. Cystic fibrosis is a good example. Everybody with cystic fibrosis has 2 not properly functioning copies of the CFTR gene. If 2 people with CF live long enough and are healthy enough and fall in love and marry and want to have babies that are genetically theirs, those babies will have to have CF. Sort of. Severity issues are a little tricky, but basically would have to have the bad genetic variations because that's all their parents have to give them. The only way those 2 parents could have a kid who didn't have 2 CF alleles is by gene editing. The second one is autosomal dominant diseases, where it takes 1 bad copy of the gene. Huntington's disease, classic example of this. It takes 1 bad copy to give you Huntington's disease. Some people are born with 2 bad copies. Most of these diseases, we don't expect those people to even survive pregnancy, to die even before birth or shortly after birth. But sometimes, in some cases, including Huntington's, in Huntington's, there's some people who are in their late teens or early 20s before they die of Huntington's disease who have 2 bad copies of the gene. They're in these villages on the west side of Lake Maracaibo in Venezuela, which is where the gene was discovered, which have a very high incidence of what's normally a 1 in 20,000 disease. If somebody who has 2 copies of a powerful autosomal dominant genetic disorder lives long enough and is healthy enough to want to have kids, the only way he or she can have kids who don't have the disease is gene editing. Those are the uses. These uses are spelled out in the National Academy report, and they say this is one of the reasons why maybe we don't want to say never do this. These people have a reason. Now, the reason would involve wanting to have kids who are their genetic kids, which isn't essential, but many, many people really want their children to be genetically theirs. So, how many such people are there? My guess is, I have no numbers on this. My guess, though, is in the United States, there might be 50 couples who meet that requirement, where both couples have the same autosomal recessive disorder, where both members of the couple have the same autosomal recessive disease, they live long enough and healthy enough to want to have kids, or they've got 2 copies of a nasty autosomal dominant. That's not going to be very common, which means there's not a lot of pressure to do this. There aren't thousands of desperate parents out there saying, I want my kid cured. I want to have healthy kids, and this is the only way I can do it. The third example is mitochondrial defects, which do pass on. If the mother has the mitochondrial disease, the child will get the mitochondrial disease. But there's a different method of getting around that called mitochondrial transfer that is currently being applied semi-experimentally in the UK and is likely to be available, clinically available and safer than CRISPR for this. So there's no strong medical reason. The reasons, though, people talk about fearing CRISPR germline babies is speciation events, the X-Men, designer babies, super babies. And yes, you could use CRISPR to take genes from, say, this is a great example because Tennille and her husband basically are superhuman. But we could make their babies even more superhuman than they already are by changing them if we knew what superhuman genes look like. And we don't. We know a lot about genes that strongly affect intelligence. And every gene we know of that powerfully affects intelligence affects it in the same direction. It diminishes it enormously. We know a lot about genes that cause diseases. We basically don't know anything about genes that make you better. The most optimistic person on the genetics of intelligence believes that he has found a genetic variation that accounts for 0.4% of the difference between child prodigies and normal kids. And other labs haven't been able to replicate that. Maybe someday we will. Maybe someday we won't, because intelligence is going to be desperately complicated. Lots, thousands of genes involved, and everything that's your entire environment, your entire life, plus chance. But right now, we don't know how to enhance kids. Maybe we will in 30 years, but this is part of my ultimate argument why we're worrying too much about it today. This is 20, 30 years off, except maybe in very limited cases. Of those 10 people a year with CF, where both members of the couple have CF, or sickle cell, or beta thalassemia, or spinal muscular atrophy, or a variety of other diseases, who want to have kids. And 20 CRISPR kids born in the world is not going to change the world. Be interesting, be useful research subjects maybe, but they don't change humanity. So those are the main practical reasons why I think we spent too much time on this. I think having a moratorium while safety issues are sorted out makes plenty of sense. I'm okay with that. But all the tons of ink that have been spilled over this have provided people's CVs with additional lines, including mine, but I don't think they're very important. Finally, I don't think it's important because if we change the human germline, so what? First, what is the human germline genome? People sort of think of it as the Ark of the Covenant. It is the code of our species. It is the essence of humanity. Okay? Well, there are approximately 7.3 billion different human germline genomes out there. Which one is the Ark of the Covenant? Obviously, it's mine, but you guys might disagree. All of our germline genomes are different. Which one is it? Further, all of our germline genomes are different from our parents' germline genomes because in my case, 24 years between when my parents were born and when I was born, the cells that ultimately became the eggs and sperm that produced me, through methods that you really don't want to think about when it's your parents, all of those went through mutations. Every time a cell divides, on average, there are about 10 different point mutations in that cell's genome, as well as uncounted other kinds of changes. Our genome, germline genome, is not static. It changes with every generation. And last, it changes in ways that are to some extent predictable. We have been changing our germline genome. First two examples. Type 1 diabetes, commonly known as juvenile onset diabetes, because it usually hits kids. Until 90 years ago, it was invariably fatal. This is the kind where you need to give yourself insulin shots or you die quickly. Invariably fatal, juvenile onset, those kids almost never live long enough to have kids that pass genes along. Turns out type 1 diabetes is not strongly genetic, but it's partially genetic. There are some genetic variations on the HLA, the human leukocyte antigen, the human immune system markers. They greatly increase your risk of getting type 1 diabetes. 10-fold, 20-fold, still below 10%, but it's a lot higher than 0.1%, right? The number of those genetic variations in our population, I'm willing to bet a lot of money on, is higher today than it was 100 years ago. Because now people who have those variations and get type 1 diabetes can live long enough to have their own kids and pass those genes on. Like my cousin Mike, who has he's not even sure whether it's 13 or 15 or 16 grandkids. He would have had no grandkids if he'd been born 100 years earlier. Medicine changes our genome. It changes our germline genome by changing the environment. Natural selection picks genomes that work in an environment. And if you change the environment significantly, you change the genomes that work in it. So the HLA variations that are linked to type 1 diabetes used to be fatal almost always before you could have kids. Natural selection is reducing those. Now natural selection doesn't act on it, so it becomes more common. In the other direction, everybody in this room has more copies of genes that help you digest starch than our hunter-gatherer ancestors. Why? Because our ancestors started farming and eating starch. And the more they ate starch, the better off people were who could digest the starch more. And so there was a small positive advantage to being able to digest starch more, a small positive advantage to people whose genomes had more of the starch digesting genes. Actually, through gene duplication, it's usually literally more copies of the gene. Our genomes have changed. They're always changing. So people talk about the human germline as the red line, the line in the sand, we shall not cross this. It was really interesting to see both Berg and Baltimore say, you know, we promised people 40 years ago that this was a line we wouldn't cross. And I said, where'd you write that down? A, I don't remember seeing that promise, but I believe they said that. And B, you promised that for you, but you're not in charge anymore. I mean, you are not science. You are not all of science. David didn't like that, actually. Times change, people change. Can you commit science and scientists forever to a promise you made? And they were uncomfortable about that, which I can understand. But in 100 years, this is going to be my great-grandchildren's world. It will be most of you, your grandchildren's world. It's not going to be our world. Their genomes will have changed. Their genomes will keep changing, whether it's changed by CRISPR or something else. I think doesn't matter in terms of whether this is a red line that we can't cross. The only argument that I see is people will say, well, it's different when we do it intentionally. And you see that argument a lot. I'm not sure, it doesn't make sense to me. If you do it unintentionally, you're doing it randomly. And who knows with what result? If you're doing it intentionally, presumably you're doing things you think are going to work. Maybe they're not going to work, but they don't have to be perfect. They just have to be better than random for it to be an improvement. It's like Tennille and I were both being chased by a bear. It's an old joke. And I say to Tennille, I don't think we could outrun him. And Tennille says to me, we don't have to outrun him. Only I have to outrun him because the bear will stop and eat me. Intentional genetic alteration doesn't have to be perfect to be better than the status quo because the status quo is not perfect. I hate to break this to you, but we are not the acme of creation with perfectly perfected, absolutely wonderful, can't get better genomes. We're muddled works in progress as a species, and that will continue to happen. And when the progress is made in part by individuals making decisions, we can look and say, is this a good decision or a bad decision? Is this a decision we want to allow or don't want to allow? My wife's hair color is now mine, except it's purple, because she's decided to dye it. She's actually had 2 people ask her if her hair color was natural. The guy who asked, she said, yes, it is. And he seemed to believe her. If people wanted to make natural purple hair color, okay, we can decide whether we think that's a good reason or not. If 2 people with CF want to have a kid who doesn't have CF, we can stop and decide, is that a good reason or not? But I don't see any reason for us to outlaw it and ban it as a matter of principle because it's a change in the germline, which is not a thing. There's a fuzzy 23,000 dimensional sphere if you only take the open reading frame protein coding parts of the DNA, 23,000 protein coding genes in the DNA. It's this fuzzy ball that's moving directions constantly. Why shouldn't we intentionally, as opposed to the way we do it accidentally through things like medicine and agriculture, change that course? However, there is an area where I think we need a lot more attention, where we're not paying enough attention, and that's the use of CRISPR in non-humans. There are a lot of constraints in doing CRISPR in humans. You have to worry about the FDA. You have to worry about tort liability. You have to worry about bad baby suits. You have to worry about public relations disasters. You have to find an IVF clinic that's willing to put its name on the line and risk this. You have to find scientists who are willing to risk it. You have to find a mother who is willing to risk it. And presumably a sperm donor, at least, who's willing to risk it. And the politics and public relations are really complicated and difficult. If you have a process that makes 100 babies and 4 of them have a severe disability as a result of the process, that's a disaster. We won't allow that. and you'll get sued, and you will go out of business, and your name will be blackened forever. But if you want to try to change mosquitoes, and you have a process that produces 4 deformed mosquitoes out of every 100, does anybody care? Actually, most people would say, why don't you go for more deformed mosquitoes? Once you move to non-humans, you don't need an IVF lab. If you're not dealing with mammals, you don't even need a veterinary lab. You don't need, for the most part, you don't have to worry about lawsuits very much. The malpractice claims for a beagle whose CRISPRing went wrong might exist, but they're going to be a lot less than the malpractice claims with respect to a baby. You don't have to find willing mothers. The practical side becomes much easier. At the same time, the regulatory system is somewhere between possibly tight, clearly loose, non-existent. With respect to animals, in the last full day of the Obama administration, the FDA issued a draft guidance, which they have not done anything with in over 12 months. The new administration hasn't confirmed or withdrawn, saying we will treat all genetic changes to animals done through genome editing as a new animal drug, which means it has to go through FDA testing, it has to go through clinical trials, it has to go through a full NDA-like process, it has to cost you millions of dollars and take you many years. Not clear to me that she could make that stick. If you're trying to change, say, mosquitoes so they can't pass malaria on to humans, is that really an animal drug? Are you really trying to cure the mosquitoes of malaria? Not necessarily. It's also not clear to me that the new administration will make it stick. Right now, that's put a damper in the US on changing animals, but there is no congressional limitation on changing the germline of non-humans. So there's a company that's trying to make cattle that don't have horns. Some breeds naturally don't have horns, most breeds naturally have horns, and the breeds that have horns, the cattle raisers burn the horns off the calves, which apparently neither the calves nor the workers like, because with the horns they'll injure each other. If you could change those genes to the gene versions that we know of from the breeds that don't have horns, you don't have to burn them off and the cattle are safe. And they're doing research under an IND, a research permission from FDA to try to get this done. There is a dog breeder in Mississippi. I've been on a panel with him. Dalmatians get particular diseases. He wants to try to cure Dalmatians of these diseases, and he's really pissed off that FDA says he has to go through this really expensive, long process, and I agree with him with respect to that. So how this will turn out isn't clear, but with animals, at least right now, there are some constraints. With plants, there are none. There are limitations on moving genes from one organism to another. These are GMOs. These are the classic GMOs. Back in the mid-'80s, something called the Coordinated Framework under the Reagan administration took authorities to a variety of agencies, EPA, Department of Agriculture, and FDA, and said, here's a regulatory scheme. 2 years ago, somebody who made a mushroom that doesn't turn brown when you cut it, they just use CRISPR to snip out the gene that causes the oxidation and the browning on the cut surface of the mushroom. These are little white button mushrooms, the boring ones that you see everywhere. Went to the USDA to get permission. USDA said, we don't have any jurisdiction over that. We don't have jurisdiction over things where you're moving a gene from one species into another species. You're not moving a gene from one species into another species. The RNA in your CRISPR construct doesn't continue to exist in this plant. We don't regulate you. On the last day of the Obama administration, USDA issued something saying, yeah, we're going to regulate them. The Trump administration has now withdrawn that. So there's no real regulation unless you're using CRISPR to move something from one species to another. But if you know that's the limitation, why bother to move it from one species to another? Just make up a new version in the species you're doing that is similar or identical to the one from the species with the gene you're interested in. Finally, as far as I can tell, there is no regulation at all in the US about CRISPRing bacteria or archaeobacteria. FDA doesn't care. USDA doesn't care. Why would you do this? Well, people are really interested in the microbiome. Maybe we're going to change people's microbiomes through CRISPR to make them better. That's not what worries me. What worries me is the guys who say, There's this virus called monkeypox. We can get samples of monkeypox. We know what the sequence of monkeypox is. There's this other virus called smallpox. Smallpox exists, we hope, only in 2 labs, one in the CDC and one someplace in Russia. But its sequence is also published. As their names might indicate, these are not very different. Or cowpox, the vaccinia that actually Jenner first used to vaccinate people. It's not that different from smallpox. You want to make a virus, you want to make smallpox, take monkeypox, take cowpox, add CRISPR, you got smallpox. That's the scariest part of this, the bio-war potential. Maybe it's 2 crazy guys with a garage. Or maybe, in the Middle East, ISIS held for over a year the large Iraqi city of Mosul. Mosul had the 2nd best university and the 2nd best medical school in Iraq. It had a modern medical school. As far as I can tell, they didn't do anything, except they did apparently execute the head of the physics department for refusing to help them build a biological weapon. Terrorist groups, individual kooks could do weird things. My Dalmatian guy, I liked him, but maybe he would end up doing something that would lead to super Dalmatians that would, wreak havoc. We don't want people to come up with new kudzu or new Australian cane toads, new invasive species that cause problems. I actually am quite pro-GMO, but I think it should be regulated some. I think somebody should look to see, is this likely to be safe? Is this likely to be too invasive? Will it do too well and move everything else out? Does it have allergens in it that people will react to? Maybe more importantly, have these people bought insurance policies or put up bonds so that if things go wrong, the people who are hurt can get some money back? Do they have ideas in place for ways to stop this? People are really excited about this for some really good reasons. So, malaria kills hundreds of thousands of people a year. Used to kill millions, it's now down to hundreds of thousands. They're mainly babies in Africa, but not entirely. Humans do not give humans malaria, except sometimes maybe by sharing IV needles, sharing needles. Mosquitoes give humans malaria. Mosquitoes get the malaria from humans and transmit it to other humans. And the malaria parasite needs to go through the mosquito as part of its sexual reproduction process. It goes through a couple of different phases. It's really screwy and weird. But if mosquitoes could not be infected with the malarial, the plasmodium that causes malaria, humans would not get malaria. People want to create, it's Aedes gambiae, I think, that passes on malaria for the most part. They want to create versions of this mosquito that are genetically immune to getting infected with malaria. And they want to spread those throughout Africa, and there are parts of Africa that are extremely interested in this, where the governments are engaged in talks, and early planning is going on to try to do this. There's a trick that I don't have time to explain called gene drive, which makes things pass through a population faster by keeping CRISPR in the next generation. So if you think about it, let's say you've got a dominant trait, and the normal wild type has two versions of the gene that allow it to get malaria. And you make a bunch of mosquitoes in the lab that have a malarial and a non-malarial, that have 2 malarial resistant genes. They mate in the wild. All the babies have 1 good and 1 bad gene. If it's dominant, they have the trait. If it's recessive, they don't. Even if it's dominant, they mate with others. And pretty soon it disappears, because it's just, unless there's a survival advantage, it gets lessened. So gene drive keeps CRISPR in the organism, and when you get that second generation that has 1 malarial resistant and 1 malarial susceptible gene, the CRISPR in it changes the malarial susceptible gene into a malarial resistant gene. And when it mates for the next generation gets changed also. In theory, you could change 1 billion mosquitoes in 10 mosquito generations, which is a day and a half. Not quite a day and a half, but it's not very long. So this needs regulation and thinking about. And we don't have regulation for it. And that's the part that scares me. Not because I think frankenfoods are terrible, et cetera. I'm worried, very worried about mistakes. I'm worried about terrorism. We need a regulatory scheme for that, and nobody except me is talking about it. That's not quite true. There's a National Academies report, which is incredibly annoyingly wishy-washy about it. But that's what needs our attention instead of wasting time worrying about super babies who aren't going to show up for 30 years, if ever. Last point, the patent fight. Some of you may know that there are arguments about who invented CRISPR. Actually, there are no real arguments about who invented CRISPR. CRISPR was invented by bacteria about 3.5 million years ago. Everybody agrees with that. But who figured out how to use it as this tool? Jennifer Davin and Emmanuel Charpentier, Jennifer from Berkeley, Emmanuel from a variety of different European institutions, published first in June of 2012 in Science. Feng Chang from Harvard, published in December of 12. George Church from Harvard, published in January of 13. A guy named Siksnis, I can't pronounce it, a Lithuanian name with accent marks that I don't understand, published in October of 2012. All of them filed for patents. Normally, first to invent, first to file or first to invent will get it. Harvard and Feng filed second, but they invoked a special patent procedure that allows them to get fast answers. It's risky, so people don't do it all the time. Feng got the patent. And UC has been trying to knock Feng's patent out. Feng is trying to knock UC's patents out. Feng and Jennifer and Emmanuel actually are quite friendly. None of this has to do with them. They don't get the money. They don't own the patents. The patent will be issued in their names because only real human beings can get patents. But if you're students, you probably already assigned the rights to any invention you can make to the University of Utah. If you're faculty, you've certainly assigned it to the University of Utah. My inventions all go to Stanford. It's the universities that are fighting over it. They're fighting in part through companies that they've licensed to use these. So one of the companies, Editas, which has the Broad Institute, Broad is half Harvard, half MIT, that's where Feng is. They spent $40 million in 1 year fighting on CRISPR patents. Part of me says, wonderful. My patent law students who graduated are not going to be able to donate more money to Stanford Law School. They're ordering the Tesla S with all the works, the ludicrous version. In Boston, their lawyers are ordering the special heated sleighs for the Boston winters. The lawyers are getting rich, or at least moderately well off. But there's no good reason to do this. This is idiotic. And every outsider says this is idiotic. And the speculation is that I've heard from some inside sources, as well as speculating myself, the head of the Broad Institute is this famous guy named Eric Lander, and who is famous for many things, including his ego. And Eric wants the Broad to be viewed as the winner, and maybe to help Feng get a piece of the Nobel Prize. So there's one thing we're worrying too much about. There's one thing we're worrying too little about. There's one thing that the fact that it's a thing at all proves that there is deep idiocy in the world, in the patent fight. These are three applications, three issues with respect to CRISPR. They are only the three that I had time for, and I think I took more time than I had anyway. There's a lot going on. This, I think, really is going to be the Swiss Army knife for biology for the next century, for at least a lot of biology for the next century. It's really interesting. It's really important. It needs people who can both understand enough of the science to be able to talk about it and understand enough law to be able to do something about it. Law and bioscience, or law and biomedical sciences, biomedicine or biomedical, is a crucial field for the future of law, of bioscience, and I think actually of humanity. So thank you for taking this course. Thank you for being at this colloquium. You now know more about these issues than 99.999% of humanity. As that noted source said, with great power comes great responsibility. It would be the first ever Spider-Man comic book. It was the narrator. People think it was Uncle Ben because it was Uncle Ben in the movie, but in the comic book it was the narrator. I charge you, pay attention to this stuff. You now know a lot about it. You got a responsibility to do something with that knowledge. Thank you for your time and attention. I'll be happy to try to answer questions.